Have you ever looked at the incredible complexity of life and wondered, what if we could speed up evolution? What if we could guide it, not over millions of years, but in a lab to create something truly extraordinary, something nature itself never even dreamed of? Well, get ready, because that's exactly what scientists are doing with a mind-blowing technique called directed evolution. Imagine being able to custom design tiny biological machines called enzymes to perform tasks far beyond their natural abilities. We're talking about superpowered enzymes that can break down plastics, create eco-friendly medicines, or even produce clean energy more efficiently. It's like building a super athlete, but at the molecular level, and it's all thanks to deliberately introducing controlled errors into DNA. So why do we need these super enzymes? Think about it. Natural enzymes are fantastic at their jobs, but those jobs are usually related to a living organism's survival and reproduction. They're great for digesting food or building cells. But what about breaking down stubborn plastics that pollute our oceans, or synthesizing complex drugs without harmful byproducts? Nature didn't evolve enzymes for those specific human challenges. Here's where the genius of directed evolution kicks in. Instead of trying to design the perfect enzyme from scratch, which is incredibly difficult, we let evolution do the heavy lifting. The core idea is simple. We deliberately introduce tiny random changes, or errors, into the DNA sequence that codes for an enzyme. Think of it like slightly changing a recipe again and again, hoping to stumble upon a much better version. How do we introduce these errors? Scientists use clever laboratory techniques like error-prone PCR or DNA shuffling. Don't worry about the fancy names. Basically, they're methods to make copies of the enzyme's DNA with a slight random chance of making a mistake each time. This creates millions of slightly different enzyme versions, each with a unique tiny tweak, like siblings with subtle differences. Now comes the crucial part, selection. We've got a huge library of these tweaked enzymes. We then set up a test, a screening process, to see which of these millions of slightly different enzymes performs our desired task the best. For example, which one breaks down plastic most efficiently, or which one produces the most medicine. It's like a massive tryout looking for the molecular superstar. Once we find an enzyme that shows even a slight improvement, we isolate its DNA, make many copies of it, and then repeat the whole introduce errors and select process again. This is the directed part of directed evolution. We're guiding the evolutionary path. Each cycle, we pick the winners, make more variations, and little by little, we steer the evolution towards our desired super enzyme, building on small improvements over generations. This is fundamentally different from natural evolution. Natural evolution is slow, undirected, and driven by survival in a specific environment. It takes eons. Directed evolution, however, is rapid, highly focused, and goal-oriented. We're not waiting for nature. We're actively pushing the boundaries of what's possible, much faster than any natural process could ever manage. A true pioneer in this field is Dr. Frances Arnold, who won the Nobel Prize in Chemistry in 2018 for her groundbreaking work on directed evolution. Her insights transformed this concept from an idea into a powerful, practical tool, proving that we can indeed teach old enzymes new tricks, or even invent entirely new tricks for them. What makes this truly revolutionary is its ability to create enzymes with capabilities far beyond what natural selection could achieve. Natural selection optimizes for the environment it's in. We, however, can create enzymes that thrive in extreme temperatures, high acidity, or with totally artificial chemical compounds, conditions that don't exist in nature, but are crucial for industrial processes. Think about an enzyme that can break down a specific type of plastic, like PET, in just hours instead of centuries. Or an enzyme that can produce a life-saving drug with 100% purity, eliminating costly and polluting purification steps. These aren't futuristic fantasies. These are real-world examples of what directed evolution is enabling right now. The power lies in this guided trial and error approach. Instead of trying to engineer every tiny detail, we create a diverse pool of options and then let the survival of the fittest principle work for us, but with a clear goal in mind. It's like finding a specific needle in a haystack, but we've built a super fast, intelligent magnet that gets better with every search. So where are these superpowered enzymes making a difference? One huge area is green chemistry and sustainability. By using highly efficient enzymes, industries can reduce waste, use less energy, and avoid toxic chemicals. This means cleaner manufacturing, less pollution, and a healthier planet for all of us. Another critical application is in medicine and pharmaceuticals. Directed evolution helps create enzymes that can synthesize complex drug molecules more efficiently, leading to cheaper and more accessible medications. It's also being used to develop better diagnostic tools and even improve gene therapies, offering new hope for treating diseases. And let's not forget biofuels and industrial processes. Imagine enzymes that can convert agricultural waste into fuel much more efficiently, or create new, sustainable materials with properties we can only dream of today. Directed evolution is a key player in building a more efficient and resource-friendly future for many industries. The future of directed evolution is even more exciting. With advancements in AI and robotics, scientists are now integrating machine learning to predict and guide these evolutionary pathways even faster and more precisely. We're moving towards a future where we can design and evolve biological solutions to almost any problem with unprecedented speed and accuracy. So directed evolution isn't just a fascinating scientific concept. It's a powerful tool that allows us to harness and accelerate nature's most fundamental process. By deliberately introducing controlled errors, we're not just observing evolution, we're actively participating in it, creating biological solutions to some of humanity's biggest challenges. It's truly science fiction becoming reality, right here, right now. What superpowered enzyme would you like to see created? Let us know in the comments below. If you enjoyed this journey into the incredible world of directed evolution, please hit that like button, share this video with a friend, and subscribe for more amazing science stories. Your support helps us bring more awesome content to you. Thanks for watching.